lipids. First off, we'll start by discussing what lipids actually are. So lipids are molecules made up of carbon, hydrogen and oxygen, like carbohydrates, except there is a lot less oxygen. They are hydrophobic, meaning water-fearing, which means they are insoluble in water. There are three main types of lipid. There are triglycerides, phospholipids, and then also steroids, like cholesterol. The main functions of lipids are waterproofing, for example the waxy cuticle on the surface of leaves, thermal insulation, for example blubber on polar bears, protection, for example around organs. Like carbohydrates, lipids are also really good for energy storage. Now we will look at one of the types of lipids known as triglycerides. So the structure of triglycerides, you've got a molecule of glycerol attached to 3 fatty acids. The bonds that attach this glycerol to the fatty acids are known as ester bonds. The reaction that joins these together is known as a condensation reaction, where a molecule of water is lost, but it can also be called an esterification reaction because ester bonds are being made. In cells, these triglycerides will form insoluble droplets, where the fatty acid tails are basically facing inward, shielding themselves from the water with their glycerol heads, because the fatty acid tails are hydrophobic. So let's look more closely at these fatty acids. There are over 70 different fatty acids. They look fairly similar, so they have this C double bond O single bond OH, which is known as a carboxyl group. And then they've got this variable hydrocarbon chain, which is represented by R. So this could just be one C attached to 3 H's, it could be a really long chain, depending on the fatty acid. But these chains can be either saturated or unsaturated. So if you have a saturated fatty acid, this basically means that there are no carbon-carbon double bonds between adjacent carbon atoms on the hydrocarbon chain. So an example would be this. As you can see, all the bonds between the carbon atoms are single bonds. And this means that the chain is very straight, so they can pack really tightly together. And this means that saturated fatty acids are solid at room temperature. And you often find these type of fatty acids in animal fats. On the other hand, unsaturated fatty acid will have at least one carbon-carbon double bond between adjacent carbon atoms in the carbon atom chain. So, for example, this would be an unsaturated fatty acid. Because of the carbon-carbon double bond, there is a kink in the chain. If there were multiple carbon-carbon double bonds, there would be multiple kinks. And this means that the chain is not as straight. Therefore, the fatty acids cannot pack as tightly together. This means that unsaturated fatty acids are liquid at room temperature, usually, and they are found in plants. Now let's look at phospholipids. So phospholipids have quite a similar structure to triglycerides. They still have that molecule of glycerol and they have fatty acids. However, they only have 2 fatty acids. The other fatty acid is replaced by a phosphate group. Now this phosphate group is hydrophilic, water-loving, compared to the fatty acid tails, which are hydrophobic. You will find phospholipids in cell membranes, which control what leaves and enters a cell. I'm just going to draw the structure of a cell membrane with these phospholipids. So as you can see, we've got 6 phospholipids here. Obviously in a cell membrane you'd have thousands. You've got this double layer known as a bilayer. Now the hydrophilic phosphate heads are facing outwards and the hydrophobic fatty acid tails are facing inwards, and this makes it quite hard for water-soluble substances to pass through, so the membrane is a barrier to those substances. Lastly, we've got the test for lipids, known as the emulsion test. So the method for this test: first you need to add alcohol to your sample. Then you need to shake it so that the lipid dissolves. Then you will need to add water. If the test is positive, a white emulsion will be produced. This is basically because in the water, the lipids will form droplets like this that will be dispersed throughout the water, and when light passes through, they will scatter the light, giving this emulsion, cloudy white effect. The more lipid that is present, the more milky the colour will appear. If there is no lipid present, then the solution will remain clear. Now pause the video and have a go at these questions. Question 1. What elements do lipids contain? They contain carbon, hydrogen and oxygen, but a lot less oxygen than carbohydrates. Question 2. What are the 3 main roles of lipids? So you've got waterproofing, thermal insulation, and also protection. Question 3. Draw the structure of a triglyceride. So you've got your molecule of glycerol attached to 3 fatty acids. What bonds join the fatty acid tails to glycerol? That's ester bonds. Question 5. What are the 2 types of fatty acid? You've got saturated and unsaturated. Saturated being the one which has no carbon-carbon double bonds, and unsaturated having at least 1 carbon-carbon double bond. Question 6. Why are unsaturated fatty acids liquid at room temperature? Because the carbon-carbon double bonds cause the chain to kink, so the molecules can't pack as efficiently. Question 7. What do all fatty acids have in common? They all have that carboxyl group on the end. Question 8. Where are phospholipids found? They are found in cell membranes. They make up the bilayer. Question 9. Draw the structure of a phospholipid. So you still have that molecule of glycerol, and you have 2 fatty acids, and then you've got a phosphate group replacing the 3rd fatty acid. Question 10. What test do you use to detect lipids? You use the emulsion test. Describe the method. So firstly you add alcohol to your sample, then you shake to dissolve the lipid, and then you pour into water. And if the test is positive, a white emulsion will be produced. Thank you for watching.